Then we move on. Next, we have Morgane Callon from MMSB in Lyon. And well, you're going to talk about line widths, I guess. Okay, then the floor is yours.
Thank you very much. And also thank you for the organizers for the invitation here. So I'm Morgane Callon. I'm working in the protein solid state NMR team led by Anja Bockmann in Lyon. And we are using solid state NMR and mainly fast magic angle spinning solid state NMR to study viruses and viral proteins. And today I will show you information on dynamics from solid state NMR spectra using an empirical approach. And I will illustrate this with our recent study of the glycoprotein GN from a virus, the Crimean Congo hemorrhagic fever virus. First, you are all familiar with solution state NMR, but maybe not all of you are familiar with solid state NMR. So I just want to give you a very brief overview. So in the solid state, we are using rotors. You see here, it's our protein containers where the proteins are sedimented. And these rotors, they are placed in the spectrometer at an angle of 54.7 degrees in respect to the magnetic field. And the idea is that if you spin the rotor around this angle, it will remove all the anisotropic interactions and in particular the dipolar couplings between protons that broaden your NMR signals, and you will obtain narrow lines. So that's why it's called the magic angle, and we are talking about magic angle spinning or MAS. And over time, the size of these rotors is reducing, and you can spin them faster and faster, the smaller they are. And now we are using rotors of a size of 0.7 millimeter diameter that we can spin at 110 kilohertz. But the question is, why do we want to spin faster and faster? And it's illustrated here. So it's two HNH spectra of the core protein of the hepatitis B virus. And you see here the spectrum recorded on a 1.3 millimeter rotor at 60 kilohertz, and here on a 0.7 millimeter rotor at 110 kilohertz. And you see that spinning faster increases your resolution and your signal. And this is because the faster you spin, the better your dipolar couplings are averaged out, the ones that broaden your NMR lines, so you obtain narrow lines and better resolution. And also, when you spin faster, you also increase your relaxation times, so you get more efficient polarization transfers between the nuclei, so you gain in signal. And actually, it exists also even smaller rotors of 0.5 millimeter or 0.4 millimeter rotors that you can spin at 160 kilohertz, and you can see here an example that you even more increase your resolution and your sensitivity. And so in the solid state, the proton line width, the one which is reduced by faster spinning, has different contributions. So the first one is the dipolar coupling, coherent contribution, which is coming from the dipolar couplings, which are not fully averaged by the magic angle spinning. And of course, you see it as it's dependent on 1 over the spinning speed. So the faster you spin, the more you reduce this contribution, the more narrow is your line, because the faster you spin, the better your dipolar couplings are averaged out. Then you have a second contribution, which is the incoherent contribution. And this is the one that contains dynamic informations, dynamic at the microsecond time scale. So it's similar than the one you have in the solution states, the R2. And these two contributions together, they are called the homogeneous line width. It's magic angle spinning dependence. Then you have another contribution, which is the exchange contribution, which is coming from a conformational or chemical exchange. But in many cases, or often, it's treated together with this incoherent contribution. This is what I will do later. And finally, you have the last contribution, which is the inhomogeneous contribution here, which come from sample inhomogeneity or magnetic field inhomogeneity. So it's independent on the mass. And you can improve it by better sample preparation or by probe design or shimming. So now the question is, so you can measure, all of these line widths or contributions. So the total line width is the full width at half maximum. Then you can measure we've seen this just before, this homogeneous contribution using a spin echo experiment where you plot the intensity decay of your isolated resonances in function of your echo time. And then by fitting it, you obtain the T2 prime, so the transverse relaxation rate. And then the homogeneous line width is 1 over T2 primes time pi. And then, of course, you get finally the inhomogeneous contribution by the difference between the total and the homogeneous line width. And if we are interested in dynamics, it's inside this incoherent contribution. So how do we access it? Because we've seen we measure the total homogeneous contribution. So how do we disentangle these two contributions? It's difficult. They are both magic angle spinning dependent, so you cannot separate them by varying your spinning speed. So one to disentangle them, you can measure the R1 rho, so relaxation times under spin lock. The idea is that in the presence of a spin lock field, the coherent contribution is strongly reduced and the decay reflects mainly the incoherent contribution. And you can measure with the spin echo experiment where the same as the homogeneous contribution, you plot the decay in intensity in function of the spin lock time, and then you obtain your T1 rho or R1 rho value. But it's still difficult to extract directly dynamical parameters from there. So what I will show you today is how you can do that by, instead of doing a theoretical interpretation, but using an empirical approach. So by comparing your data with model systems with known dynamics. And I'll show you this on the example of a viral protein, so the glycoprotein N from the Crimean Congo hemorrhagic fever virus. And just before going to details, I just wanted to introduce briefly the virus we are working on. Actually, this virus is on the WHO priority list because it has a high potential to trigger future pandemics, because it has a fatality risk that can go up to 40%, and today there is no treatment nor vaccines. And it's part of a larger class of viruses, which are called Bunia viruses. It has more than 450 members, and a lot of them have a very high zoonotic potential. And actually, this virus is emerging in Europe because the tick that is the vector of this virus, the yellow tick, is moving northwards due to climate change. And just as a comparison, this is a tick which we are familiar with, this forest tick that transmits the Lyme disease. And this yellow tick is bigger. It has stripes. You cannot see it well here, but it's bigger. And it has eyes. And actually, it can hunt you. And you can see that it's really following someone, which is here. So it's really actively chasing you. I'll stop this video. So actually, it's already in Europe. Reported cases were reported in almost all Europe, also in UK. And actually, I checked this morning, only one human case in the UK, but it was imported case. There is no indigenous case. In Spain, there is already indigenous case, and already people died from the disease. So if we go to the virus here, you see a scheme of it, and you see that in the envelope there is two glycoproteins. The first one is GN, so it's a glycoprotein N, which is made of an ectodomain, a transmembrane domain, and a cytosolic domain between the two TM helices. And this protein is recruiting the viral RNA for the viral capsid reconstitution. And actually, from this protein, only the structure of the isolated cytosolic domain is known. It was solved by solution state NMR, and you see it here. The quality of the figures are really poor from the beginning. I'm really sorry about that. So hope you see it. It's a double zinc finger, and actually each zinc finger is made of 3 cysteines and 1 histidine coordinating a zinc atom. There is also another glycoprotein in the envelope. It's a glycoprotein C, you can see here. And actually, really recently, the structure of a virus from the same class, the Andes virus, which is infamous now because it's a cruise ship virus that you all heard about. So the structure of GN and GC was solved, and it was shown that it forms a heterodimer that oligomerizes into a tetramer. It was solved by EM. And what is really interesting here is that you see that the cytosolic domains were not visible, and probably because they are dynamic. And it's what is interesting us here. So we studied it, and we started with a part of the GN protein, the cytosolic domain and the transmembrane domain, which we call GNTM cyto. And we studied it and the dynamic of the cytosolic domain. All the biochemistry was done by my colleague, Marie-Laure, Caroline, and Laura, and the NMR and the relaxation data were recorded at ETH Zurich by Susanne Penzel, a former PhD student, and all was supervised by Anja Bockmann. So after we prepared the protein, we obtained the first H&H spectrum here, you see, of the protein. And what you see that it's just a big blob, meaning that there is a lot of overlaps. And using this spectrum, it would be impossible to collect site-specific relaxation data or dynamics. So we need to improve resolution to be able to measure such kind of data. So to improve the resolution, we used selective labeling of amino acids. So only the selectively labeled amino acids will be visible in the spectra, which will improve the resolution. And we'll be able to measure all the dynamic parameters we wish to. And if you produce your protein in bacteria, you can selectively label amino acids, for example, the methyl groups of the isoleucine, leucine, and valine, or the alanine or methionine, but it's not working for all of them. And here is why. So here you see the metabolic pathway map in E. coli. And it shows how all the amino acids are connected. And for example, if you want to label some amino acids as alanine here, phenylalanine here, you could do it easily because they are at the end of the path. But for example, if you want to label threonine, you see it's connected to many other amino acids. So if you label threonine specifically, your labeling will end up all over. And this is called scrambling. And to avoid this scrambling in the lab, we are using cell-free protein synthesis to produce our protein that I will call CFPS. And the idea is that instead of using cells to produce your proteins, you mix all the ingredients necessary in a pot. So the energy source, the ribosome, the amino acids, the coding RNA. And because you directly add your amino acids, you can really choose the labeling scheme you want here. And also what is really interesting is that the scrambling is very limited. You see it, it's limited to this. And you can even use enzymes that block these reactions. So you can really label what you want. And this was developed by, in the lab by my colleague Marie-Laure. That even improved it. The only problem, let's say, is that the yield is really low. You get about 1 milligram of protein per prep. But now, thanks to the small rotors we use in solid-state NMR, the 0.7 millimeter rotors, this is compatible because now we need less than a milligram of protein to study it. And you see that 10 years ago, using the 3.2 millimeters, you would need 30 milligrams of protein. So it would make impossible the use of CFPS to produce the proteins. But now this is entirely feasible. So we selectively labeled our protein. So we selectively labeled the cysteines, the glycines, and the phenylalanine. And you see the resulting spectrum in blue here. And you see that now we improve the resolution and we can really detect single resonances. And actually, if we overlay it with the solution state spectrum of the isolated cytosolic domain of GN we recorded, you see that we could even transfer the assignment to these resonances. And we could start measuring relaxation and dynamic parameters. So that's what we did on the different assigned resonances. You have them here. And we, as I said at the beginning, we used an empirical approach. So we compared our results with model systems, which were recorded under the same conditions, same magnetic fields and mass rates, and all the proteins are deuterated and backprotonated. So we compared it to ubiquitin, which is a mix of alpha helices and beta sheets, ASC, which is a fully alpha helical protein, and ETS, which is made of beta strand and from fibrils. And this was recorded by Susanne Penzel at ETH Zurich. And if we start with the total line width here, we can clearly see that for our protein, the total line width is the larger. And this is what we observed already in the spectra, that we had really broad peaks. And if we move to the inhomogeneous contribution, you see that this is the main contribution to the line width. So clearly, and this is a bit similar to what is shown in ETS, and clearly the main source of broadening is the sample inhomogeneity. And now if we want to go deeper, we can compare the homogeneous contribution, and we see that first, it's 2 to 3 times higher than for our model systems. And you remember this homogeneous line width has 2 contributions, the coherent contribution and the incoherent contribution. And the coherent contribution was coming from this not fully averaged out dipolar couplings between protons. So it depends on the proton distances. And in alpha helices, the distance between amide protons is shorter than in beta strands. So the coherent contribution would be larger for alpha helices. So the homogeneous contribution. And actually, this is what we see. If we zoom on ubiquitin, we see that it's larger for the alpha helical part and smaller for the beta strands. And also clearly in ASC, which is fully alpha helical, we have larger homogeneous contribution than in ETS, which is fully beta strand. So what it means is that when comparing similar secondary structures between proteins, the differences we see in the homogeneous line width relate to the presence of motion. And actually, this is what we see for GN, that when we compare similar secondary structures, we have higher homogeneous contribution than for the model proteins. So there is the presence of dynamic in GN. And to go further with this, we measured this R1 rho, so the relaxation rate under spin lock. It's shown here in blue, and we compared it also to the R2 prime that we measured in pink. So first, in blue, if you check on model proteins, R1 rho don't have this dependency to secondary structure. So you see it's similar in helices or beta sheets. But what is interesting is that when we have fast R1 rho and R2 prime, here around residue T9 in ubiquitin, around residue 36 in ASC, or 246 in ETS, all these regions are dynamic. So both fast R1 rho and R2 prime are indicative of flexible segments. And if we do this comparison in GN, we see that for these 2 residues, 772 and 780, they both show long R1 rho and R2 prime, indicative of dynamic. And they are both part of the second zinc finger of the protein. So the second zinc finger is dynamic. And in the contrary, for example, for this residue 756, we see that it has a long R2 prime, but a very short R1 rho, which indicates that it's a rigid residue. And actually, it's in the first zinc finger, which is then rigid. So overall, this analysis showed that the sample inhomogeneity is the main contribution to the line width, but dynamic is present, and the second zinc finger here is dynamic. So as a take-home message, I wanted to show you that the proton line width in solid-state NMR contains different contributions: the coherent effect, so from the dipolar couplings, the incoherent effect that contains the dynamic information, but also sample and field inhomogeneity. And that by measuring these contributions, but as well R1 rho, you can access to microsecond timescale dynamic. And by comparing it, your data with well-characterized model proteins, you can interpret dynamic and understand a bit better the dynamic in your system. So with this, I'm at the end, and just a quick slide to thank my team at MMSB, our collaborators and former postdoc lab at ETH Zurich, and thank you very much for your attention.